All right, 9 o'clock Central Time. Good morning, everybody. My name is Lorena Moscardelli. I'm the PI of STARR. And this is the BEG Technical Seminar on our Fridays. It's with great pleasure that I want to introduce Vann Smith with Ellington Resources. Vann is a biostratigrapher specializing in nanofossil and palynological micropaleontology. He got his bachelor's degree from Louisiana State University on geology and geophysics. He also has a master's degree in earth and environmental sciences from Tulane University and a PhD from Louisiana State University. In 2020, he continued as a postdoctoral researcher and geology instructor in Louisiana State University. And in 2022, he was hired by Ellington Geological Services as a staff biostratigrapher. He has a wide range of interests on his research that include end Cretaceous mass extinction, early Paleogene hyperthermal events, Cenozoic plant evolution, and the reconstruction of Holocene climate using pollen records. I'm really excited today to introduce Vann, who is going to give a presentation where he will be showing some results from a STARR Ellington collaboration that we initiated last year in the onshore Lower Wilcox Group. So with that, I'm gonna pass the baton to Vann. Vann, the floor is yours.
Okay, thank you, Lorena. I will share my screen. So can everyone see this?
Yes, it's working.
Okay, cool. Okay, good morning, everyone. First, I'd like to say that it's an honor to be invited to be a speaker for the Bureau. And I hope you find the talk interesting. We have some great collaborators here. I'm going to be mainly focusing on the palynology, but there's also a lot of work being done on the chemistry, the geochemistry, and the lithostratigraphy and other aspects of this research into the Lower Wilcox Group. So a brief introduction to our research team. You guys probably know most of these people already, but I wanted to just mention them briefly. So Dr. Lorena, who introduced me. We have Dr. Yulia, Evan Sybil, all at BEG STARR, Dr. Hessler at the Deep Time Institute, and myself, Tanya Lorente, and Kudri Liu at Ellington. And Rich Howe has done some work for us with the nanos as well. So the purposes of this study, we are looking at 2 subsurface wells in the Lower Wilcox Group onshore. And as a palynologist, I had a particular interest in doing some alpha taxonomy, which basically just means the description and describing how we name different species and that kind of thing. And I'll explain that in a little more detail later. We are also interested in stratigraphic correlations for the Lower Wilcox, both biostratigraphic, chemostratigraphic, lithostratigraphic, and also to compare it with previous work done on outcrop, onshore, and the deep water. So there's important applications here in oil and gas exploration. The deepwater Wilcox is an important hydrocarbon reservoir, and palynology is often used for the biostratigraphy, and particularly because it's often nanofossils and forams are not as useful just because they're diluted. And also has important implications for energy transition, like carbon capture and storage. And it also has implications for paleoclimate. There's been a lot of previous work done in the Lower Wilcox, including by some of the collaborators on this project. that I'll briefly mention. So this map here is showing the delta system in the Wilcox, and then the Wilcox outcrop. So it outcrops sort of parallel to the coastline here, and then dips down into the subsurface to the southeast. Just to briefly mention a few, we have Olario and Ambrose looking at growth faults in the Guadalupe Delta. Angela Hessler has done work on continental weathering. Olario and Zing looking at deltas in the South Texas coast. And there's a lot of other STARR research projects. This study is going to focus on a core and cutting samples from the Jerome, Olinick, and Jay Simmons-1. I also wanted to briefly mention the previous talk that you guys had at December 2nd, which would be a great thing to look at, in addition to this, covering sort of different angles of this and looking at a bit of the younger Wilcox, the PTM section. So to give a bit of geologic background, this is a Cenozoic, showing the Cenozoic in Texas. And so after the end Cretaceous, you have deposition of this Midway group, which is mainly shales and open marine. And then you get a big influx of plastic material related to the Laramide orogeny and depositing some very thick sandstones. That means it's an important hydrocarbon reservoir as well. And this is a outcrop of the Wilcox from Shreveport that I saw back in 2021, and you can see some of the I'm including this map just to give you guys a general idea of what the paleogeography looked like around the time of the deposition of the Wilcox group. So we have some of these deep water systems here, and then delta systems, and the Laramide orogeny here. This is also showing some PTM localities, and I'll briefly mention my dissertation research was on looking at the PTM section in this Chicxulub core. So previous work in palynology in the Wilcox group. As far as outcrops onshore, a lot of people have published things going back to the 1960s. For the onshore subsurface, there's a paper by Gregory and Hart in 1995 looking at subsurface Wilcox from Louisiana. And more recently, there's a paper on the offshore subsurface by Zara et al. 2019 with Rebecca palynologist there, and did a really good job coming up with a biostratigraphic zonation for the Wilcox here. And this work is going to be looking at the subsurface Lower Wilcox onshore Texas in Karnes County. These are the locations of the two wells here. They're right next to each other in Karnes County. And this is just showing where they are superimposed on a geologic map of Texas. You can sort of see the outcrops getting older as you go northwest, paralleling the coast. So let's talk about the palynology. And in case you're not familiar with these groups, I'm going to go give sort of a short background. So these are examples of different types of pollen that we're seeing in the Lower Wilcox. The size range here is for palynology in the Cenozoic is generally 10 to 200 microns, some rarely bigger than that. Then we have nanofossils.
Vann,
yeah.
Sorry to interrupt. Can you get closer to the microphone? I think when you turn your face to the right, we lose a little bit of sound.
Oh, sure. Sorry about that. Yeah.
Thank you.
Let me get the screen a little closer. Is this better?
That's good. When you do that, it's good. Yeah.
Okay, cool. Yeah. And so nanos are smaller than palynomorphs. They're also calcareous, and forams, the kind that we look at in biostratigraphy anyway, are calcareous, and they range from less than 100 microns to over 10 centimeters. In some cases, they can get huge. These are individual single-celled organisms here, bigger than a quarter. So what exactly is palynology? Most people think pollen immediately, and that's very important, but we also study lower plant spores, plant tissues, fungal remains, dinoflagellates, algae, animal fragments, acritarchs, and we just lump those together and use a term called palynomorph, which just means any organic walled microfossil. People have Traverse has described it as what my net catches is a fish. So anything we see in the palynological preparations is fair game for palynology. And an important thing to consider in palynology is that we want to figure out the biological affinities of these fossils, and not just like stamp collecting. We want to figure out what they're telling us about the paleo environment. So here's some modern pollen types and lower plant spore type from my Holocene research, and then comparing it to similar types from the Lower Wilcox. So you can see that, you know, we have sphagnum, pine trees, and alder trees here, and the pollen looks similar. And when I mentioned alpha taxonomy, I just wanted to give this example. So what do we call this pollen? There's been a bunch of different names applied to this species, and people have had arguments about whether some of these are synonymous. And so part of what I'm doing is sort of going through the naming of it and trying to make it clear what I mean when I'm calling it this, and if it's potato or potato, right? So the methods, we use the standard acid technique using hydrochloric acid and hydrofluoric acid to process the samples and put them on microscope slides for me to look at. This is done at Global Geolab in Canada. And I won't go into detail here, but the idea is you want to get rid of the carbonates and silicates, and you're just left with the organic fraction. And I did quantitative abundance counts here. I won't go into detail about the methodology there, but I can if people have questions. So the samples that I have so far are 31 samples from the Jay Simmons and 48 samples from the Jerome Olinick number 16. And we're going to be getting more samples, but analysis has been completed for palynology for these wells. So this is the kind of output you get in stratabugs when you graph all of these pollen types. I've simplified the taxonomy here a little bit and grouping things together into genera instead of every species. Otherwise, this would be unreadable. There'd be like 100 different types, right? So this is the biostratigraphic interpretation based on palynology, sample depths, self-potential. This is total palynomorph abundance. And then this shows the relative abundance of different types. So the dark green here is terrestrial pollen and plant spores. The blue is dinoflagellate cysts. And you can see that they're all mainly terrestrial pollen and plant spores that have been transported. So all the samples are dominated by angiosperm pollen. We've identified 204 total types, 142 pollen and lower plant spores, 36 dinoflagellate cysts, 12 algae, 14 fungal bodies. And these will all be described. This is well over 23,000 individual palynomorphs counted for these two wells. So let's talk about the biostratigraphy a bit. There's two very common types of biostratigraphic events would be your top and your bottom. So this is showing this pollen group, Omnippides caryopollinides in Wyoming, and using this as biostratigraphic zonation to split up the Paleocene. This one species, Triorbicularis, has a very short range. So when I say bottom, I just mean the lowest stratigraphic occurrence. Top, I mean the top. And people have different terms for this. I like using top and bottom. And then there's also abundance changes can be events here like the highest common occurrence. And I won't get into more detail here. We do have to consider reworking and caving in biostratigraphy. So reworking is when a pollen gets eroded and redeposited. So you might, if you don't know it's reworked, you might think you're older than you actually are. Sort of like a dinosaur fossil ending up in the Paleocene. Caving is sort of the opposite problem that you have with well cuttings, where younger fossils can fall down into lower samples. And we'll go back to this zonation in a bit, but this is sort of zoomed into the zonation that STARR has that I'm basing this biostratigraphy on. So looking at Jay Simmons-1, at the top of the well, it is either Selandian or Phoenician. We're in Paleozone 4. There's this local marker that isn't in STARR, but is I thought was important because it becomes quite abundant. So it's a very clear event at the top, possibly useful for regional biostratigraphy. There's the base of P. McGregori indicating a Selandian age. Then the base of common Caryopolynides ripides. And then the base of Caryopolynides ripides, which is a little uncertain because these are cutting samples and they could be caved. And it's possibly top Danian at the bottom sample. Looking at the Jerome Olinick number 16, again, Paleozone 4 at the top. The base of P. McGregori. Again, that's local marker Monipides annulus. These events are quite close to each other, and they're actually in flipped in stratigraphic order. So they're pretty close in age, and analysis of more wells will help clarify which one is truly above the other. We again have base common Caryopolynides ripides. There's a big sample gap here due to poor expected poor palynomorph recovery with sands. And then base Caryopolynides ripides. And the bottom sample we think is still Selandian in age based on this dinoflagellate Areolagera medusetiformis complex. And we recently have some initial results from the some nanofossil analysis of the Jay Simmons well by Richard Howard Ellington. These are very preliminary. He's just looked at the top and bottom samples of the Jay Simmons well so far. And in those slides, it's very low abundance, 1 to 2 specimens per slide traverse at 1,000 times magnification, and it's low diversity. Which sort of can make it more difficult to get a more precise biostratigraphic age on it. There's a Phoenician aspect for the top sample based on the presence of these species, and no evidence of Precambrian or early Eocene taxa. So, and the bottom sample could be as old as Upper Danian with Prinzius martini and Tueus pertussis. This is definitely worth further study. It is probably going to be quite time consuming if the other samples are as rare as this one. But it's nice to see that the initial results are consistent with the palynology and lithostratigraphy. And just to show you what these nanos look like, here's a couple of pictures of them from the Lower Wilcox. Okay, let's talk about the paleoenvironmental interpretation. So this is a nice cartoon by Valley et al. 2020, showing what happens to pollen after it leaves the plant. So pollen can be transported by wind and water. It can end up in rain and snow. So the pollen that we're seeing in this shallow marine area is a mix of local and long distance transport and fluvial transport and wind transport. So for the paleoenvironmental interpretation of these pollen and plant spores, there's two approaches. The first is nearest living relative. So what is, you figure out what is this pollen, what affinity does it have, what family or modern genus is it, and what environments do they live in today. This becomes more uncertain the older you get back in time. And then there's also the paleogeographic and paleoenvironmental distribution. So if you are looking in the Cretaceous and you see a pollen type and you only see it in the paleotropics, then it's, indicating tropical conditions, that kind of thing. And so I wanted to show you some pictures of the pollen types that I'm seeing, and also pictures of modern plants that we think are pretty similar to what the plants look like that produce these pollen in the Paleocene. So a very common type is what we call this Betulaceae miraceae type, which includes several genera, but it might have looked something like the southern wax myrtle with similar environmental preferences. Momippides and Caryopollinides are very common, and they're related to hickory trees. Some of the other more common pollen types, the affinity is less certain. So Nyssa pollinides, despite the name Nyssa, it is somewhat uncertain, but it may be related to modern water tupelo, which is sort of a lowland forest marshy type tree. Thompsonopolis magnificus, this is probably even more uncertain, but it is quite common. Maybe relationship with the Rubiaceae. This is a button bush from Texas. Some of the less common types, we have palm tree pollen of this tropical tree, kapok tree, similar to it. Pollen from water lilies and elm tree pollen. Then for the gymnosperm pollen, we have some pine pollen. We have this extinct group called Classopolis that I'll talk about in a bit. Pollen related to modern ephedra, which generally lives in arid conditions, and pollen from that may represent something similar to bald cypress, although some other groups can produce it. For the lower plant spores, these are the more common types, well, except for this one. I included this one because it's interesting. We can be pretty confident that this is a tree fern pollen, Cyathea. Deltoidospora could be from a lot of things, but maybe it looks something like this golden leather fern. Levagatosprides, maybe another fern, like something like this common polypody. And then Stereosporides steroides is basically sphagnum, which generally indicates moist conditions. I've also identified the fungal spores here. They're generally not as biostratigraphically useful as the pollen, plant spores, and dinoflagellates, but that might be partially due to the fact that people haven't really studied this as much as the other groups. And so there's a lot of sort of types where I'm not too sure about the lower biological affinity, but there were some interesting types that I'm pretty sure are microtheriaceae. These live on plant leaves and sort of eat decaying plant leaves. So you can see modern ones here. And this is the Lower Wilcox version. We're also seeing freshwater algae, and most of these freshwater algae types are related to the modern algae family Zygnomataceae. So that's a lot of pictures, but the sort of big picture is that they generally indicate probable nearby terrestrial paleoenvironments, a lot of them indicating either like a warm temperate to tropical lowland forest, swamp, salt marsh, and lake environments. So the marine paleoenvironmental interpretation, the dinoflagellates were not as common as the pollen and plant spores, and often they were they weren't very diverse, not as well preserved, but the most common type was this Operculodinium type here. And this is an illustration from Slew et al. 2005. So these blue arrows and boxes indicate that we're seeing them in the core. So we're seeing genera that indicate neritic conditions generally, and some of them maybe even restricted marine. And what we're not seeing are these groups that are more oceanic. So the assemblage is indicating shallow water. And also probably normal marine salinity most of the time. And then the next thing I'll talk about is the geochemical integration. And these analyses were performed by Angela Hessler at DTI and Evan Civil at BEG STARR. This shows the Pali data together with the geochemical data. The geochemical data is very high resolution sampling, as you can see. There are various proxies that we have that I won't go into detail with, like grain size, weathering, humidity, 2 salinity proxies. So I wanted to compare that with some of the relative abundance data to see if there was a correlation. So we took a 5-point average around the palynological sample depths and then just compared the values to see if there was correlation. So this shows some of the correlations between the Pali and the geochem. To our knowledge, this is the first study which has tested the paleoclimatic utility of palynological abundance data by comparison with geochemical proxies. And it has a potential application in high resolution biostratigraphy for like 10 to the 5th order climate cycles. Some of the groups that were worked particularly well, deltoidospora versus humidity has a positive correlation. So as expected, deltoidospora, we think that it is more common in moist conditions. So the positive correlation is expected and pretty good. Classipolis versus salinity is also fairly good. And this is an extinct group, but it's thought to have lived in arid and are like salt marsh coastal habitats. Looking at it versus humidity, it's actually not well correlated in the opposite direction because we would expect it to be more common in more arid environments. So it sort of indicates Classipolis is more of a salt marsh indicator than a aridity indicator. Looking at the DS ratio, so this is the ratio between marine dinoflagellates and terrestrial pollen and spores. Generally, you expect if you see more dinoflagellates that you have more of a marine influence, but I'm not really seeing a relationship with salinity. So it may be that the DS ratio is telling us more here about the amount of fluvial input than the salinity directly. And we're going to look at 2 additional wells. So we're going to do the same thing and see if we see the same trends here. So the conclusion, well, first, there's potential high resolution biostratigraphic correlations we could make and sort of climate cycles we could look at with the assistance of this relative abundance data. So we took deltoidospora and this is just showing a comparison when you graph this aridity or humidity proxy with the deltoidospora abundance. And you can see the trends match up pretty well. This is zooming in on like 10 to the 5th order cycles here that are tracking together pretty well, which is encouraging. Another important thing to mention is this group Classipolis. So it's pollen produced by an extinct conifer family called the Chirolepidiaceae. And it's widely thought to have gone extinct at the end of the Cretaceous along with the dinosaurs. But there's evidence summarized by Berry in 2022 which indicate survival of the group into the Paleocene in South America and China. And he provided several possible explanations for their eventual extinction, possibly competition by angiosperms. And this is a map from Barry showing in green the locations where he's confident that there's Paleocene occurrences of this group, and then a question mark over North America. And so I think in our cores, this is Classopolus, by the way, we see fairly high relative abundances of this consistently present, and there's a rarity of other reworked pollen from the Cretaceous. This all suggests that the Classopolus is in situ and is a Paleocene refugium. And Barry also mentioned that these were sort of living in arid coastal salt marsh environments. So we're interpreting this as a late refugium for this extinct conifer family. This is a comparison of our results with a zonation, an earlier zonation by Elsick and Krava from Wilcox outcrops. And so I'm just paying attention to the lower Wilcox here. So this type may be useful, this Caria species, less than 30 microns. Presumably he's talking about Caria Polynides here, not the modern genus. I didn't observe any of these scalariform sieve plates. And this Massiopola Polynides species is undescribed, so I can't really be certain what they mean by that. And Momippides dilatus top also appears to be good because we're seeing it throughout our samples, and our samples are almost probably exclusively lower Wilcox with maybe a little bit of middle Wilcox and Danian at the top and bottom. So let's compare it with the deep water zonation by Zara. So some of these groups, the species over here that are not in boxes are either nanofossils or forams. These are forams, these are nanofossils. So we're not looking at those. I'm just going down through this column and saying which of these events worked and which didn't. So at the top here, Insul Polynides rugulatus, we didn't actually see the top here, but it is consistent with what we've seen. We do see this species in the core. We just don't see the top. We didn't observe these abundance changes in Momippides clearly, and Momippides actinus appears to be absent. This may be because we're getting a more regional signal from these onshore cores, and the deep water, you're seeing a broader pollen source area, and Momippides actinus is living elsewhere. We didn't observe this dinoflagellate species, but our dinoflagellates were fairly rare and not super diverse. So that's not too surprising. P. McGregori was a very useful event. We have this local marker, T. Momippides analyst, that fits right in here, maybe a little bit above P. McGregori. The next two wells should clarify this. We see Virippides as a useful event. We didn't observe these abundance changes, and we didn't observe these lowest occurrences of Pectiodinium homomorphum and multispinosum. Again, these are dinoflagellates, so it appears like the dinoflagellates are not working quite as well. I only saw one specimen of a Pectidinium, and it wasn't one of these species in the core. C. Virippides, the bottom of this, is a good event. And we didn't observe actinus in this well. And so we have some interesting results from these two wells, and I'm really excited to further work on this. We have more work to do with integrating this palynology with lithostratigraphy and the geochemistry. And we're going to be looking at 2 more wells, so stay tuned. And I want to say thanks for your time, and I look forward to any questions you might have.
Thank you, Anne. That was excellent. So we're doing great on time, and if there are any questions from the audience, please don't be shy. Any questions or comments?
This is Rebecca Hackworth. I have just a comment for Vann.
Sure.
Hi, Vann. It doesn't surprise me at all that you're not seeing the actinus. It's when we get in the deep water, I notice as we go different parts of the Gulf of Mexico, actinus is more prevalent. So there may be, some localized presence of it. So you may not be seeing it. So it'd be interesting if you get more onshore cores material to find out, what areas onshore you're seeing actinus versus, where you're not seeing it. And the analysis, also, we use that quite often. It just wasn't on the chart, so I'm glad you found that one useful.
Cool. Yeah. Well, I'm glad to hear that you're not too surprised by that absence. I was definitely on the lookout for that. And the other two cores we have are not quite as close together as these, so it'll be interesting to see if that species pops up.
Yeah, very, very much so. It's good that people are doing some onshore work in this section, because there's definitely not a lot of onshore work done. A lot of the times because that, these intervals are cooked further down dip in a lot of those cores. So I'm glad you got some good recovery. The recovery looks fantastic.
Yeah, there's really good preservation and pretty much all of the samples. And abundance was good. Yeah, it worked well.
Yeah, I think we were a little bit nervous when we were collecting the samples first, but we have been surprised in a good way with the recovery. So thank you, Rebecca, for your question. I think Sue Hovorka has a question. Sue?
Hi, I enjoyed this talk very much, Vann. Thank you very much. I'm not a Wilcox or a palynology expert, so it was really clear and illuminating for my purposes. I wanted to know about, if you could talk about what the palynology tells you about the changing environments from sand-rich intervals to mud-rich intervals. Do you collect this where do you collect your samples? Out of the reservoir intervals, or you have these also? I mean, all the different muds from the interfluvial kind of soils to marine incursion, and between delta marine incursions. So what's represented in your set, and can you reflect on especially I'm interested in the environment of the fines, the mud rocks.
Mm-hmm. Yeah, so before I started on this project, Carlos Santos was associated with it, and he did some of the sampling. He did the sampling of the core and picking the well cuttings to use. So we sort of avoided the coarser sand intervals because generally they don't have good preservation. So we're focusing on these like more mud-rich intervals. And I think one of the next things that we want to do here is really look in detail about the depositional environments versus the assemblages that we're seeing. But from the samples that have been selected, I'm not seeing big changes in the assemblage and relative abundance. So there's not like some very distinct difference between some samples and others. There's some species that slowly come in and out, and cyclic variations, which may be climatic or may be more controlled by the like whether it's a overbank deposit or different types of that. But that's something that we are definitely going to look in more detail at.
You're not seeing evidence of big marine incursions in going far up dip. You're staying mostly in the fluvial near shallow marine environment.
Yeah, well, we think it's fairly shallow, but the fact that we're seeing dinoflagellates in all of the samples, and we're also seeing like rare nanos, indicates that it's pretty marine.
Hmm.
It's not like I hesitate to say with too much certainty because I don't want to step on the lithostratigraphy and the earliest possible interpretations, but it looks to us like shallow marine and but not super deep water.
So I want to know about the continuity of we want to know about the continuity of these units. So anything you guys have to say about that tells us whether you would think they're continuous over miles, many miles. The lateral continuity of those mud units is high value to our purposes. So it's critical information. Thanks.
Yeah.
Thank you, Sue. I have a comment on that. That's part of the motivation with this work, because especially in this area, we're in this growth fault region, and it's very hard to make correlations between different fault blocks and also along strike. I think with these two wells that Vann was looking at also, we're looking at one point because they're very close to each other. I think there is a whole lot more work that could be done, kind of like spreading the sampling along strike and along deep. So, I mean, the other thing is that beyond the age control and the paleoenvironmental interpretations, Vann counted around 27,000 grains. Vann, is that correct?
Well over 23,000. It sort of varied, and there's like an estimation, so that's a minimum. Yeah. So
the value of this also is that it allows you to use relative abundances, and I think Dr. Laurent is in the audience, and we have been talking about using that as well to try to use it as a correlation tool if possible. It will tell us several things about paleoenvironments and age and everything, but also if we do it right in different sections, it could be a powerful tool for correlations, So yeah, as Vann said, stay tuned. We'll see how that goes. But there is another question by Richard then. Richard.
Hi, good morning, Vann. Good to hear some paleontology bandied about here. One comment, like on the one thing especially with Richard Howie, is when I was working at Exxon, we were doing a lot of work in it'd be a slightly younger stuff, the Vicksburg Frio play, and a lot of that was counting nanofossils and tracking. You could track really well with the sequence strat. It went really well with the sequence strat, tracking just the nano counts and seeing these bigger incursions, and then those were your good drapes, like what Sue was asking about. On the geochemistry, was that off of cuttings?
No, that was only from the core.
Okay, that's good, because otherwise you're burying like yeah, is that real or not? But okay, that's good. And I guess the other question, I saw Neil Campion is on the thing, so whether or not you have talked to them, since they've done Petrostrat's done an awful lot of work in the offshore environment, which I don't know if they've published any of that in any kind of form or not, but I mean, that's what at Marathon, that's what I relied heavily on was their work in combination with chemostrats, geochemical zonation. The two of them worked in tandem very well in tracking these units around, but obviously that was in a much muddier environment than what you're dealing with.
Yeah, well, our idea is that we think it's good that we're publishing this in sort of an open way, so that it's not like a black box, right? So I'm trying to describe everything and be very transparent here, and so hopefully we can get some more industry data into the public domain, so we can get the academics involved with this biostratigraphic zonation.
Was there anything, I mean, although it looks like the BOEM has shut that down, they used to publish the actual data for a while. Because I would suspect then some of the data that was collected may have gotten into the open, but whether that's it, so it may still be out there somewhere where, even though it was Petrostrat's data, it may be public.
Oh yeah, if I remember correctly, I think it's like 10 years and then it becomes public and you can request it. That's definitely an option to just like try to dig out all the publicly available BOEM pally data and do a zonation that way, use RASC. It's the same, it's a similar thing to what I did with nanos in the Miocene for that SEPM paper a couple of years ago.
Yeah, I mean, because I know Marathon, well, we drilled a 35,000 foot well that, you know, we had multiple people looking at, so that data may or may not be out there, but it's that's just another avenue to look at, but you're trying to figure out how the big pulses, mud pulses, they're tracked to what you've got. Anyway, look forward to it.
Something to, great comment, Richard, something to, you know, dig into a little bit more. But one of the plans for us also is all this information. We are planning for a BEG report of investigation, you know, so hopefully by the end of the year we'll have a draft in really good shape. And the intent is to make these things available for researchers and other people to kind of, you know, connect the dots and get something more robust. Okay, so are there any other questions or comments from the audience? Don't be shy. We still have probably 10 minutes. Nope. All right. Well, Vann, thank you very much. That was an excellent presentation. And I guess, you know, we will continue the collaboration. So for the audience, if you're interested on this topic, stay tuned because we will keep going with more updates from our research as we find new and exciting things. So thank you, everybody. Have a wonderful weekend. Bye.
Thanks, everyone. Have a good day.
Bye. Thank you, Vann. Excellent.